afternoon. Gordon, PhD scholar working multi-omics applications on alcoholic fat. A recent publication published in Nature Genetics, which have omics approaches in non-alcoholic fat. A brief introduction about non-alcoholic. As all you know, NAFLD is a condition where the fat infiltration is more than 5% and the secondary without identifiable secondary causes. As a spectrum, NAFLD NASH is related cirrhosis is the secondary most common cause of indication of liver transplantation. We see the prevalence is in general population increasing dramatically, increasing the global burden. Further, although the known risk factors for NAFLD are metabolic syndrome, age, hypertension, hypertriglyceridemia, obesity, and diabetes, it is important to know that genetic factors are influencing pathogenesis of NAFLD. And the changes in the it is challenges it is in challenges in both diagnosis and stage of NAFLD, where the liver images, sorry, the ultrasound can identify the NAFLD, but has a poor sensitivity. Further, the liver enzymes are commonly elevated, but they have a non-specific and poor predictive for the progression. Although the liver biopsy is a gold standard method, but it is involved in variability and risk of complications. It currently there are no pharmacological application therapies approved for the NASH, and it is a complex disease where it need identify where the need of identification of potential drug targets and biomarkers it is a paramount. So with this background, the present study aimed to uh, aimed a multi-omics approach to identify the genetic variants which are associated with the NAFLD risk factors. This study involved five different population: United Kingdom United Kingdom Biobank and the Iceland Decode study and FinGen study, Copenhagen Hospital Biobank study, and USA Whole Genome study. These are the different clinical anthropometric parameters included in this study. And the liver phenotype is assessed by the guidelines given by ICD-10 code K1796, and they are also involved in they also included 51 phenotypes and the blood biomarkers and the anthropometric markers. Further, they analyzed the meta-analysis using logistic regression for the variant and the phenotype association. As I was saying, it is a multi-omic study where they where they did a whole exome sequencing and RNA-seq analysis and proteomic analysis. Whole exome sequencing is done in blood samples using Illumina iSeq, and RNA-seq is done on Illumina iSeq, where they did RNA-seq both in blood samples and the adipose tissue samples. Further, they did a proteomic studies using a SomaScan proteomic analysis, which is a high throughput adapter based proteomic assay, and the Olink proteomic analysis, which is a 96 plus immunoassay that enabled high throughput detection. For proteomic analysis, they use the plasma sample. Further, they did a data analysis. For data analysis, Mendelian randomization was done using a R package. Further, the pathway analysis was done on cis and trans PQTL, that is a quantitative trait locus analysis for the identified variant with association with the proteins. Further, they did a liver stage classification, and they analyzed the proteins based on the stages of the NAFLD spectrum. For this, they did a logistic regression model, and for predictive score, they did a ROC and AOC curve analysis. They estimated the proton density fat fraction using image resonance MRI images, and for this they acquired two techniques. One is GRE and IDL. GRE is a gradient estimation, multi echo estimation, and there's another technique IDL they have used. Uh, they these are the settings they use for the imaging technology. The comparison of proton density fat fraction of two techniques, the two techniques difference they compared, and they observed that uh, there is a no difference. Both techniques found the same similarity, showing the fat fraction in the both cohort. Here we can see out of 29,000, the fat fraction, that is NFL, identified in very less population. Further, they compared the correlated the fat fraction and the BMI. Here we can see the BMI is positively correlated with the fat fraction, and they did a fraction part. They did a comparison for high versus low BMI PDFF in UKB cohort. They identified only 2% of high PDF and low BMI, and further they identified 19% of individuals having a high PDFF and high BMI. With high BMI, these results show that the BMI increases results in the increase of fat fraction in the individuals. They did a GWAS analysis where they done in a PDFF estimated population, and further they analyzed the meta GWAS analysis in clinically diagnosed NFL patients. The variants associated with combined PDFF and NFL at genome-wide scan were nominally significant both for both the phenotypes. So they combined the both phenotype. These GWAS identified 18 variants in 17 loci were associated with only non-alcoholic fatty liver, whereas the two additional variants identified were associated with cirrhosis, not with PDFF. Further, they done a potential causal candidate gene analysis. For this, they analyzed the 46 million sequence variants, where they measured the mRNA levels and evaluated the lead of affected amino acids and the mRNA expression. For this, they did a QTL analysis. These analysis results in 14 variants which were in high LD with missense variants. Further, they identified a missense variants in these four genes which are associated with corresponding gene mRNA expression, and they also identified a top SQTL, that is splice QTL analysis results showed variants in these three genes, and they also identified an intronic variant in TOR1B gene in exon 2 upstream region. It is associated with the cryptogenic splicing, which results in the truncated protein. These are the identified 18 variants in non-alcoholic fatty liver disease. Here we can see the strongest association between the PNPLA3, TM6SF2 missense variants, which are highly associated with the phenotype. Further, they did a meta GWAS analysis to identify four in a cirrhosis patient. For that, they identified four variants in these four genes which are associated with only cirrhosis, not with NFL. And they did a effective identification variant on PDFF compared with the effect of liver disease and liver enzymes and lipids. Here they identified the PDFF effect except variants in ADH1 gene and the HFE gene, all are associated with the disease progression, and the same results were observed for liver enzymes, and with the lipid profile. They did an association of NFL variants with the other triad and the outcomes. This heat map shows the association between the variants and the identified 51 variants. Here we can see most of the variants were associated with the liver enzymes and NFLD spectrum. Further, we can see variant in MTRC1 gene has a greater similarity in their association with the tested traits. A variant in ApoH is associated with increased risk of arterial fibrosis and heart failure and higher level of lipoprotein. A variant in ADH1 is associated with reduced risk of alcohol consumption, and most of the variants were associated with cholesterol and the other parameters. This is the forest plot showing association pattern of missense and loss of function variants in GPAM and mTOR-C1 gene, and here they found the association of the selected phenotype was similar with the uh, missense variant and loss of function variants. Further, they did a variant and variant BMI interactions. There was a interaction between splice site variant in HSD gene and the missense variant in PNPLA3 gene are associated with reduced level of ALT levels, even AST. These interaction of these two variants diagnosis of NAFLE and SCC also significant. That is, it is associated with the risk of the disease. Further, the 20 NFL variants were screened for the interaction. Only they could see the difference between difference for the variants in ApoE and HSD gene with the increasing BMI. A nominal significance was observed for the missense variant in PNPLA3 with the increased BMI, and they also identified a missense variant in PNPLA3, TM6SF2, and ApoE and GUSB, which is interacted with BMI to affect the PDFF, that is increasing the fatty infiltration in the population. They also analyzed the circulating protein levels. For this, they analyzed the protein levels measured with the 4907 aptamers by the SomaScan technique and 1459 immunoassay using O-link technique. Further, they analyzed the protein levels with the identified 20 variants, and where they could find the association for the 16 variants only for in trans proteins. Further, the trans PQTL analysis identified two variants in GCKR and SAPNA1 genes, which are associated with the other proteins. And they also identified 273 proteins which are associated with the variant traits which are in trans. They identified a missense variant in PNPLA3, which is the top trans QTL for the 86 proteins. This is the heat map showing the effect of identified variants with the proteome. And missense variant in MTRC1 and GPA1 were associated with level of group X11B sector phospholipase A2 lipase protein, which is highly expressed in the liver and associated with non-alcoholic fatty liver disease, which was previously known and associated with the NAFLD disease. And they also identified the most significant difference between NFL and cirrhosis for the two proteins, which is one is IGFB P2 and 7, which are highly expressed. Further, they did a ROC model for trained to discriminate between the NFL and cirrhosis. The trained logistic regression models used liver enzymes, age, sex, BMI as the baseline, and the models all discriminate between NFL and cirrhosis. Further, the combined plasma protein and genetic risk scores is a better than the genetic risk score. However, the genetic risk score can identify early onset of disease and the individuals, so the identified individuals for the study summarizes the GWAS identified 18 sequence variants in NAFLD disease and 2 variants in associated with the cirrhosis. Further, the longitudinal PDFF measures suggested that the missense variant carriers in PLNPLA3 gene were more susceptible than the body mass index, susceptible for the body mass index than non-carriers, suggesting that they also suggested that the GPAM1 and MTRC1 have a essential role in inhibition of GPAM1 and mTORC1, and these could be a therapeutic for NFL and NASH. They also done a plasma protein analysis, which revealed that the missense and predictive protein loss variants were associated with the PLA2 GX11B in plasma. And they also find the association of IGF P2 and 7 with the cirrhosis and NFL is inconsistent with the studies identified. With this study conclude use of multi-omics data to shed light on the genetic basis of NAFLD, analysis of protein loss of function variants and blood mRNA expression, plasma proteomic data pointed the causative genes and changes in their functions contributing to pathogenesis. Further, they demonstrated that the derived effect of NFL risk allele on the other disease trait, including blood lipids and proteins, showed that the plasma proteomics has the potential to stage the NFLD. These are the limitations in the study. There is a lack of enabling more detailed phenotype stratification, in particular to histological data for the disease staging. Information on the other causes of the liver disease, such as alcohol consumption, is limited. However, the approach was in line with the recent opinion to not base the disease diagnosis on a measure of exclusion of other diseases such as ALD. Thank you.
Afternoon, everyone. I have been given the task of interpreting this paper that is more than just presented and a comment on the paper. So whenever we read such an elegant study, the first thing that comes to mind is how does this fit our kind of a setting? So from a geneticist perspective, there are two very important points that we look at. One is the sample size, and the second, the ethnicity in which it was conducted. There's no doubt that this is a very huge sample, so there's no problem there. But if you look at this, one of the study populations that is quoted here is only people of the white background have been included in this study. So this is one of the so-called limitations of the study, that it has been carried out in a particular ethnicity. Now, most of the genome-wide association studies that were initiated during early 2000s, this has been the problem, and a lot of experts have been commenting that there's no genomic diversity in the human genetic study. Now, the problem is if you were to correlate this particular data to any of the other ethnicities, it would be a problem. The other thing is there's a lot of talk about the benefit of the genetic studies to precision medicine. So again, this lack of diversity in the study, probably this would hamper the application of this particular studies to precision medicine. Now, is this really a problem? I'll just give you two examples from the lab that we have recently seen that ethnicity plays a major role, and the way that you interpret a particular variant changes altogether from a particular ethnicity to a different ethnicity. Now, this is one of the landmark papers that was published in New England Journal of Medicine for two reasons. One thing, in the absence of an approved therapy for NASH, this was one of the gene targets that was likely to be targeted in future for NASH. And the second is the whole approval for targeting this particular gene is a natural way because there's a splice variant in this particular gene and it leads to absence of the protein, and that absence of the protein actually causes some kind of a protection. Now, if you see the abstract, the results, there are three very elegant points that the study has highlighted. One is the variant is associated with reduced levels of liver enzymes. The second is it confers not only protection to alcoholic liver disease, but also to non-alcoholic liver disease, heterozygotes by 17%, and the homozygotes by almost around 30%. The third and most important thing, as Govardhan has just presented, the top hit is a variant in a gene called PNPLA3 that confers risk for fatty liver. And this particular variant mitigates the effect of PNPLA3. What does that mean? That means that if an individual has a variant in a PNPLA3 gene, he has susceptibility to fatty liver. However, if the same individual has this particular splice variant, he is protected from fatty liver even though he has a risk variant. So these are the three elegant points that this paper has summarized. Now, this is of course a landmark study. So we initiated the study at our lab to see whether the same variant confers protection in the Indian population. I will summarize the entire study. If you look at the A, when we looked at the liver enzymes between splice variant carriers and non-carriers, there was no significant difference in the levels of the enzymes, suggesting that probably in the Indian population, this variant is not affected with the liver enzymes. The second one, what we have looked at is we have taken the PNPLA3 normal and HSD normal individuals versus PNPLA3 variant and the HSD variant. So what, according to the NEJM publication, was the variant PNPLA3 carriers and the HSD carriers should have lower liver enzymes. But what we see is in our population, in fact, they have higher liver enzymes. That is, whoever has a PNPLA3 variant and a HSD variant, they have higher variants. Now, in whom this is pronounced, to look at that, what we have done is we have taken obese patients with wild type and variant, and we have also taken lean. So what we are seeing here is only in the obese patients who have a PNPLA3 variant and a HSD variant, they have higher liver enzymes, suggesting that probably in a subset of patients in this combination, we'll have to be careful because they might progress fast. Now, why do I say that? I say that because when we compared all the clinical and the other parameters between HSD wild type and variant carriers, what we see is there is no significant difference between any of the clinical and demographic characteristics, but only the VLDL is different, where the levels are low in the HSD carriers, and low levels of VLDL actually suggests that they are at a higher risk of progression to the higher etiology. So again, any patient with a PNPLA3 variant and a HSD variant are more likely to progress fast in our ethnicity. Now, we actually demonstrated that both PNPLA3 and TM6SF2, both the variants which are the top hits in the current Nature Genetics paper, back in 2014, we have shown that these also play a role in the Indian population. However, if you look at this cartoon, the color summarizes the prevalence of NAFLD in the general population, and the light blue color indicates the frequency of risk variant in the general population, right? So if you look at, say, for example, the Hispanic American, almost 50% of the population have the risk variant in PNPLA3. Whereas if you look at the Indian population, less than a quarter of them actually have a variant in PNPLA3, which is the predominant driver of the disease in the Western Caucasian population. It's at a very low level in the Indian population. So this gave the impetus to us that we should start our own study. So that's what we have done. Now, whenever we do a genetic study for most of the common diseases, there are two ways that the genetic susceptibility actually causes the disease. One is the common variants that actually cause the disease. The common variants are, they are at a higher frequency in the general population, like the variants in TM6SF2 and PNPLA3, or the rarer variants which have a frequency of less than 1% in the general population. These are very rare and probably limited to a particular subset of patients or a particular subset of ethnicity. So that's what we tried to address when we did a whole exome sequencing study, where we sequenced the entire exonic regions and the untranslated region in 37 NASH patients who are either biopsied, or they have persistently elevated liver enzymes, or they have a family history. And then we have also taken the controls where there was no fatty infiltration. We did the whole exome sequencing and then did the analysis. Very quickly, what we have seen is, we wanted to see whether it is the rare variant which is actually initiating and driving the disease, or the common variants like the PNPLA3, TM6, or any other variant which are common in the general population, but because of some kind of environmental interaction are actually initiating the disease. Here, interestingly, almost 70% of the patients had a very rare variant in them that actually probably initiated the disease. If you see on the left hand side, you have pathogenic variants with a minor allele frequency. That's the definition for a rare variant, a minor allele frequency of less than 0.01% in the general population, and a medium frequent pathogenic variant which has a minor allele frequency greater than 0.01%, but then it is a pathogenic variant. And the third, variants that are already reported in literature, something like the TM6SF2, PNPLA3, and almost around 101 variants that are already reported in the general in the literature. So what we see is more or less the number of variants that are already reported are more or less the same across all the 37 patients. But if you see the number of rare pathogenic variants, they differ in each of these patients, and almost 70% of the patients had a rare pathogenic variant. Again, the number doesn't mean anything, it's the function that they are involved in. Again, very quickly, the controls had none of the pathogenic variants in the so-called lipid or the other genes that typically initiate the NASH. Now, if you see, if you were to summarize, most of the patients had either a rare pathogenic variant in a cholesterol lipid metabolism, or a adipose obesity related autophagy, circadian rhythm, or all the phenotypes that are very essential and reported in literature to actually initiate the spectrum of NAFLD. And we see that almost 70% of the patients have a rare variant in them. If you were to compare both of this, if you see the variants reported in literature, the down panel is the control and the top panel is the patient samples. But for the rare pathogenic variants or common variants which are pathogenic, you see rest of the variants are more or less similar across the entire stretch, meaning the entire study group, meaning the controls or the patients. Now, if I were to summarize these findings, the rare variants, if you see, the reported whatever 18 loci that have been reported, the paper that you have seen, none of them are here. So it's a different rare pathogenic variants that are probably seen in our population. Say, for example, one on the X chromosome, probably there's some kind of male this thing, because one X is a variant, it's enough to start the disease in the male. So probably that male preponderance might be explained here. And then of course the LGR4, where there is altered lipid homeostasis because of the rare variant, and then antioxidant enzyme SOD3, and then the CERKL. ATG7 is one of the variants which is involved in autophagy, which is already reported in the literature for initiating NASH. So we have seen that there's a gene called CERKL, which is a regulator of autophagy, and again a rare variant in this actually probably is involved in initiation of the disease. To summarize, yes, elegantly done study, multi-omic approach, but we need more of ethnic diversity in the studies that we do, and probably we need our own studies to actually so-called identify the Indian variants which are actually involved in the disease, and the precise role as to how they initiate or probably protect from the particular. Thank you all for a patient
My actual talk is actually clinical applications of this particular study, but I just want to brief upon what is multi-omics, because I am a pure clinician and I am poor in this omics part. So, everybody is well aware of omics, but for us, when we are sitting in a patient, we are a little unclear. When I got a message saying that I have to speak on clinical applications, then I went to for 48 hours, I brainstormed myself for what is actually omics. So the central dogma of cell, any cell, basically it's from DNA, it transcripts into RNA, and this translated to protein. This was said around decades back, and this has been followed, and we only knew these parts for long, till 2000 probably. Then the study of this DNA is called as genomics. The study of RNA is called as transcriptomics. The study of protein is called as proteomics. This is the simplest thing. Then came the epigenetics, the study of factors that control the DNA through DNA, either through mRNA or methylation, is called as epigenetics. And when the proteins are metabolized, it is called as metabolomics. So this was the basic thing which we should understand as a clinician. I see many of the scientists, I think everybody knows this, but still it was new to me. The study of all the omics is called as multi-omics. When they integrate everything and translate into a clinical output, then we say it is a useful multi-omics study. So systems biology is a field which was developed in 2000, in the year 2000 probably, which was initially the institute was set up at Seattle, and then Harvard came up with an institute saying systems biology is quite important. And in the past 2 decades, we have seen significant progress in this field. So it is a field of study, particularly the study of interactions between the components of biological systems and how these interactions give rise to the function and behavior of that system. So when we understand these systems, all what the study here mentioned here, that there are certain proteins which are expressed, genomes which are found. Although I feel that PNPLA3 and TM6SF2 has been common between alcoholic liver disease and fatty liver, because we have reviewed the data and see that many of the patients, in fact, there is the UK Biobank, the same study was published in Journal of Hepatology saying that PNPLA3 and TM6SF2 increase the risk of alcoholic cirrhosis as well. Patient consumes alcohol a significant amount. The patient is similar two groups, one who has expression, one who doesn't have the PNPLA3. The one who has the PNPLA3, they have higher risk of cirrhosis despite consuming similar amounts of alcohol. So this is the basic network and approaches in the omics layers. The first two is for assessment of the for diagnosis and the mechanisms, and the later ones for diagnosis and prognosis. It has been applied for prostatic cancer significantly. They identified one or two markers. One is sphingosine, which was identified. What they do for all the omics, we need tissue. Without tissue, it's quite difficult, especially for genomic study, as I understand from the studies whichever I went through, that you need a tissue to say that whether there is a abnormality in the cancerous tissue and a normal tissue. And when there is when you compare these two and compare the metabolomics, you get some marker which can say whether this is proven to predict cancer or not. So the patient of prostatic hyperplasia has a normal prostate specific antigen, which we say is a tumor marker for prostate. The patient has normal PSA as well. Sphingosine is a metabolite. If you check that if the sphingosine is too high, then probably patient has more favorable towards cancer rather despite having a normal PSA, prostate specific antigen. So this was one marker which was quite useful for identifying prostatic cancers. Second thing, for HCC, I've taken two examples. For HCC, there is a new drug which has come, atezolizumab plus bevacizumab, quite rapidly used everywhere in India and US, is the first line choice. But the moment it came, the study initially said that it works very well for viral hepatitis, but not for NASH. Then there was a Nature paper saying that the NASH, this is because of the tumor microenvironment where there is increased expression of CD8 and T1 cells, and that's the reason that these drugs, immune checkpoint inhibitors, do not work in this group. Then they did a multi-omics study which was published in Hepatology, quite interesting study, and has been discussed in all the wide forums as well. The multi-omics identifies the link between intratumor steatosis and the exhausted tumor immune microenvironment in the heat hepatocellular carcinoma. Initially, they took a tissue, non-viral HCC tissue. They said that there are three classes, class 1, class 2 and class 3. Class 1 and class 3 is quite important. Class 1 has TP53 mutation and class 3 has CTNNB1 mutation. So if the patient has class 3, is here as shown in the figure, survival is too good. Patient has survival of more than 5 years, the patient receives the drugs. And the TP53 mutation has very poor prognosis. So this first they identified the genes, then they identified the tissue microenvironment, and they said that patients who have steatotic type of tumor, they have increased the expression of CTNNB1 and lesser incidence of lesser amount of TP53. There is increased expression of immune class as well. But the immune microenvironment, although the cells are there, they are not active. So when you give a drug to them, the patient responds very well. From tissue, then they identified the protease marker, which I have not discussed here. And then they said that steatosis is a marker of good response to HCC. For identification of again steatosis, just they did MRI for the next patients and found that patients who had good amount of fat in the HCC, called a steatotic HCC, they respond very well. So from lab to simple MRI can diagnose that yes, just they initially diagnosed, this was quite cumbersome to take 113 patients with tissue, then analyze and then say that patient has very good response to drug, and then do a simple MRI, and next patient will be saying that okay, this patient has steatotic HCC, he will respond very well to drug. So this is the simplest applications of this clinical application of multiomics. But problem with multiomics, what we feel is, one, we require tissue from every patient. It's quite useful for diagnosis, useful for prognosis, useful for treatment response as well. For NAFLD, there have been multiple studies. This is one of the nature studies, was groundbreaking study, which said again that PNPLA3 and TM6SF2 has been increased. And there is a genetic risk score, GRS, which is based on these two and one more gene which is added to that, HSBD beta 13, the one which was discussed. And they said the genetic risk score can determine whether the patient can develop steatosis or can progress to cancer. But HCC progression to cancer has not yet been proven, but there are only few studies which say that yes, patients, if they have this, may have increased risk of HCC. But NASH is the leading cause. That there are several trials which are undergoing, and multiomics are still need to be done. Anything else to be added, Ravikant? Oh, cancer. Otherwise, issue. Okay. Thank you. Thank you.
And also do metabolic.
Huh.
It's circulation, plasma. So
But if there are multiple metabolites in the blood, how will you confirm that yes, this is the metabolite which is responsible for the disease or association?
We also do
So all these, because I was also thinking the same thing. PNPLA3 is just the association. Many of us might have PNPLA3 but may not develop fatty liver.
Yeah.
80% may develop, but 20% are still cannot have.
Is not a related food.
That's true.
And
So there is a database to say that this protein is related to